All right, our topic for this week's module are going to be nucleic acids. Specifically, what we're going to focus on this week is going to be DNA, deoxyribonucleic acid. This is going to be chapter 10 in your textbook, all right? Just for reference, there's more in chapter 10 than just DNA, all right? Chapter 10 is kind of a dense chapter, so we're going to divide it into two separate weeks. This week, we are just going to focus on the structure of DNA, how DNA was discovered, and how DNA replicates, okay? In a couple weeks, we'll go over the next kind of steps in chapter 10. We'll cover transcription, which is going to discuss RNA, translation, which will cover protein, and we'll also talk a little bit about mutations and how that relates to DNA, okay? But for today, what we're going to focus on in this learning module, we're going to focus just on DNA. So if you remember, way back when we started this class, I drew up a nucleotide, okay? And that nucleotide is the individual monomer that makes up DNA. It also makes up RNA. But what we have that makes up that nucleotide are going to be three components, and I'm going to remind you of those three components now. So we've got a nitrogenous base, we've got a sugar, and we've got a phosphate group, okay? Now remember, when we're talking about DNA, we've got to pay attention to what's on what carbon. So we've got a 5-carbon sugar here, and I'm going to number the carbons with the pink marker. So the first carbon is where our nitrogenous base is always attached to. With DNA, that's either an A, T, C, or G. So adenine, thymine, cytosine, guanine, those are our bases associated with DNA. We've got our sugar. Okay, our sugar, that's an A, is deoxyribose, okay? And we know it's deoxyribose because at that 2' carbon, we've got a hydrogen. And this is going to be important, okay? Having that hydrogen at that 2' carbon is what allows our double helix to form, okay? We've got our 3' carbon here. This is also going to be an important structure. This is going to be where the next nucleotide would be added in making our polymer, okay? Because remember, monomer is the individual unit, the monomer is the nucleotide, the polymer is going to be the DNA strand. That's going to be lots and lots of nucleotides strung together, all right? And that's where our next one's going to happen. Our 5' carbon, that's where our phosphate group is added. It's not added, sorry. That's where our phosphate group is located. That would be more accurate for me to say, okay? So we need our phosphate group. We need our sugar. And we need our nitrogenous base. That's going to be what makes up our nucleotide. Okay? And really, DNA, when it was discovered, a lot of the early research was using viruses. Okay? And the reason for that is because viruses are a lot simpler than eukaryotic cells. Okay? Viruses consist of nucleic acid and protein. And this was known way before the actual structure of DNA was discovered, okay? Based on that virus being made up of nucleic acid and protein, scientists knew that one of these two macromolecules had to be the genetic information that was passed on from parent to offspring. And what happened, all right, and there's a video in the PowerPoints this week that I'd like you to watch. Two scientists, Hershey and Chase, did a series of experiments where they labeled the nucleic acid with one radioactive isotope. They labeled the nucleic acid with P32, or phosphate 32, okay? That's the part of the molecule that got labeled. The protein was labeled with S35, or sulfur 35, okay? And by tracing where the P32 ended up and where the S35 ended up with the particular bacteria that they used, okay? And the bacteria, they used, not with the bacteria, with the particular virus that they used. Sorry, the virus they used was a bacteriophage. And you'll learn more about bacteriophage in microbiology, but bacteriophage is a virus that specifically infects bacteria. So what Hershey and Chase did with their experiment, they radioactively labeled P32, they radioactively labeled the bacteriophage with P32, or they radioactively labeled the bacteriophage with S35. They then used those radioactively labeled bacteriophage to infect bacteria. And through a series of experiments, they figured out where the radioactive material ended up, okay? So in order for that macromolecule to be the genetic information, that radioactivity had to end up in the bacteria, okay? And what they observed with their experiments is the P32 ended up in the bacteria. The S35 was outside, so it didn't get into the bacterial cell. What that told them, and what was really groundbreaking and neat about that experiment, is they definitively showed that DNA with the labeled P32 was the genetic information, okay? So viruses were actually very important in the early studies of DNA. Your textbook has a little bit of background on viruses, but what I want you to focus on specifically are the experiments with Hershey and Chase, and there's a little animation in the PowerPoints for you to watch, okay? If you have any questions, please let me know.